Regulation of Respiration, Chapter 126. Introduction. Respiration is a reflex process, but it can be controlled voluntarily for a short period of about 40 seconds. However, by practice, breathing can be withheld for a long period. At the end of that period, the person is forced to breathe. Respiration is subjected to variation, even under normal physiological conditions. For example, emotion and exercise increase the rate and force of respiration. But the altered pattern of respiration is brought back to normal, within a short time by some regulatory mechanisms in the body. Normally quiet regular breathing occur because of two regulatory mechanisms, nervous or neural mechanism. Chemical mechanism. Nervous mechanism. Nervous mechanism that regulates the respiration includes respiratory centers. Afferent nerves. Efferent nerves. Respiratory centers. Respiratory centers are group of neurons, which control the rate, rhythm, and force of respiration. These centers are bilaterally situated in reticular formation of the brainstem, Fig 126.1. Depending upon the situation in brainstem, the respiratory centers are classified into two groups: medullary centers consisting of dorsal respiratory group of neurons, ventral respiratory group of neurons, pontine centers, apnostic center, pneumotaxic center, medullary centers, dorsal respiratory group of neurons. Situation. Dorsal respiratory group of neurons are diffusely situated in the nucleus of tractus solitarius, which is present in the upper part of the medulla oblongata, Fig 126.1. Usually, these neurons are collectively called inspiratory center. All the neurons of dorsal respiratory group are inspiratory neurons and generate inspiratory ramp by the virtue of their autorhythmic property, Table 126.1. Function. Dorsal group of neurons are responsible for basic rhythm of respiration, see below for details. Experimental evidence. Electrical stimulation of these neurons in animals by using needle electrode causes contraction of inspiratory muscles and prolonged inspiration. Ventral respiratory group of neurons. Situation. Ventral respiratory group of neurons are present in nucleus ambiguous and nucleus retroambiguous. These two nuclei are situated in the medulla oblongata, anterior and lateral to the nucleus of tractus solitarius. Earlier, the ventral group neurons were collectively called expiratory center. Ventral respiratory group has both inspiratory and expiratory neurons. Inspiratory neurons are found in the central area of the group. Expiratory neurons are in the caudal and rostral areas of the group. Function. Normally, ventral group neurons are inactive during quiet breathing and become active during forced breathing. During forced breathing, these neurons stimulate both inspiratory muscles and expiratory muscles. Experimental evidence. Electrical stimulation of the inspiratory neurons in ventral group causes contraction of inspiratory muscles and prolonged inspiration. Stimulation of expiratory neurons causes contraction of expiratory muscles and prolonged expiration. Pontine centers. Apnostic center. Situation. Apnostic center is situated in the reticular formation of lower pons. Function. Apnostic center increases depth of inspiration by acting directly on dorsal group neurons. Experimental evidence. Stimulation of apnostic center causes apnosis. Apnosis is an abnormal pattern of respiration, characterized by prolonged inspiration followed by short, inefficient expiration. Pneumotaxic center. Situation. Pneumotaxic center is situated in the dorsolateral part of reticular formation in upper pons. It is formed by neurons of medial parabrachial and subparabrachial nuclei. Subparabrachial nucleus is also called ventral parabrachial or colliker fuse nucleus. Function. Primary function of pneumotaxic center is to control the medullary respiratory centers, particularly the dorsal group neurons. It acts through apnostic center. Pneumotaxic center inhibits the apnostic center so that the dorsal group neurons are inhibited. Because of this, inspiration stops and expiration starts. Thus, pneumotaxic center influences the switching between inspiration and expiration. Pneumotaxic center increases respiratory rate by reducing the duration of inspiration. Experimental evidence. Stimulation of pneumotaxic center does not produce any typical effect, except slight prolongation of expiration, by inhibiting the dorsal respiratory group of neurons through apnostic center. Destruction or inactivation of pneumotaxic center results in apnosis. Connections of respiratory centers. Efferent pathway. Nerve fibers from respiratory centers leave the brain stem and descend in anterior part of lateral columns of spinal cord. These nerve fibers terminate on motor neurons in the anterior horn cells of cervical and thoracic segments of spinal cord. From motor neurons of spinal cord, two sets of nerve fibers arise, phrenic nerve fibers, C3 to C5, which supply the diaphragm. Intercostal nerve fibers, T1 to T11, which supply the external intercostal muscles. Vagus nerve also contains some efferent fibers from the respiratory centers. Afferent pathway. Respiratory centers receive afferent impulses from peripheral chemoreceptors and baroreceptors via branches of glossopharyngeal and vagus nerves. Stretch receptors of lungs via vagus nerve. By receiving afferent impulses from these receptors, respiratory centers modulate the movements of thoracic cage and lungs through efferent nerve fibers. Integration of respiratory centers. Role of medullary centers. Rhythmic discharge of inspiratory impulses. Dorsal respiratory group of neurons are responsible for the normal rhythm of respiration. These neurons maintain the normal rhythm of respiration by discharging impulses, action potentials, rhythmically. These impulses are transmitted to respiratory muscles by phrenic and intercostal nerves. Inspiratory ramp. Inspiratory ramp is the pattern of impulse discharge from dorsal respiratory group of neurons. These impulses are characterized by steady increase in amplitude of the action potential. Impulse discharge from these neurons is not sudden and it is also not uniform. Inspiratory ramp signals. To start with, the amplitude of action potential is low. It is due to the activation of only few neurons. Later, more and more neurons are activated, leading to gradual increase in the amplitude of action potential in a ramp fashion. Impulses of this type discharged from dorsal group of neurons are called inspiratory ramp signals. Ramp signals are not produced continuously but only for a period of 2 seconds, during which inspiration occurs. After 2 seconds, ramp signals stop abruptly and do not appear for another 3 seconds. Switching off the ramp signals causes expiration. At the end of 3 seconds, inspiratory ramp signals reappear in the same pattern and the cycle is repeated. Normally, during inspiration, dorsal respiratory group neurons inhibit expiratory neurons of ventral group. During expiration, the expiratory neurons inhibit the dorsal group neurons. Thus, the medullary respiratory centers control each other. Significance of inspiratory ramp signals. Significance of inspiratory ramp signals is that there is a slow and steady inspiration, so that the filling of lungs with air is also steady. Role of pontine centers. Pontine respiratory centers regulate the medullary centers. Apnostic center accelerates the activity of dorsal group of neurons and the stimulation of the center causes prolonged inspiration. Pneumotaxic center inhibits the apnostic center and restricts the duration of inspiration. Prebot-Singer complex. Prebot-Singer complex, PREBOSC, is an additional respiratory center found in animals. It is formed by a group of neurons called pacemaker neurons, located in the ventrolateral part of medulla. Pacemaker neurons generate the rhythmic respiratory impulses. Medullary centers send nerve fibers into this complex. Exact functioning mechanism of this complex is not known. Factors affecting respiratory centers. Respiratory centers regulate the respiratory movements by receiving impulses from various sources in the body. Impulses from higher centers. Higher centers alter the respiration by sending impulses directly to dorsal group of neurons. Impulses from anterior cingulate gyrus, genu of corpus callosum, olfactory tubercle and posterior orbital gyrus of cerebral cortex inhibit respiration. Impulses from motor area and sylvian area of cerebral cortex cause forced breathing. Impulses from stretch receptors of lungs, Herring-Brewer reflex. Herring-Brewer reflex is a protective reflex that restricts inspiration and prevents overstretching of lung tissues. It is initiated by the stimulation of stretch receptors of air passage. Stretch receptors are the receptors which give response to stretch of the tissues. These receptors are situated on the wall of the bronchi and bronchioles. Expansion of lungs during inspiration stimulates the stretch receptors. Impulses from stretch receptors reach the dorsal group neurons via vagal afferent fibers and inhibit them. So, inspiration stops and expiration starts, Fig 126.2. Thus, the overstretching of lung tissues is prevented. However, Herring-Brewer reflex does not operate during quiet breathing. It operates only when the tidal volume increases beyond 1000 ml. Herring-Brewer inflation reflex and deflation reflex. The above-mentioned reflex is called Herring-Brewer inflation reflex since it restricts the inspiration and limits the overstretching of lung tissues. Reverse of this reflex is called Herring-Brewer deflation reflex and it takes place during expiration. During expiration, as the stretching of lungs is absent, deflation occurs. Impulses from J receptors of lungs. J receptors are juxtacapillary receptors which are present on the wall of the alveoli and have close contact with the pulmonary capillaries. As Paintal discovered that these receptors are the sensory nerve endings of vagus. Nerve fibers from these receptors are non-myelinated and belong to C-type. Few receptors are found on the wall of the bronchi. Conditions when J receptors are stimulated: pulmonary congestion, pulmonary edema, pneumonia, overinflation of lungs, microembolism in pulmonary capillaries. Stimulation by exogenous and endogenous chemical substances such as histamine, halothane, bradykinin, serotonin, and phenyldigmanide. Effect of stimulation of J receptors. Stimulation of the J receptors produces a reflex response, which is characterized by apnea. Apnea is followed by hyperventilation, bradycardia, hypotension, and weakness of skeletal muscles. Role of J receptors in physiological conditions is not clear. However, these receptors are responsible for hyperventilation in patients affected by pulmonary congestion and left heart failure. Impulses from irritant receptors of lungs. Besides stretch receptors, there is another type of receptors in the bronchi and bronchioles of lungs, called irritant receptors. Irritant receptors are stimulated by irritant chemical agents such as ammonia and sulfur dioxide. These receptors send afferent impulses to respiratory centers via vagal nerve fibers. Stimulation of irritant receptors produces reflex hyperventilation along with bronchospasm. Hyperventilation along with bronchospasm prevents further entry of harmful agents into the alveoli. Impulses from baroreceptors. Baroreceptors or pressoroceptors are the receptors which give response to change in blood pressure. Refer Chapter 101 for details of baroreceptors. Function. Baroreceptors in carotid sinus and arch of aorta give response to increase in blood pressure. Whenever arterial blood pressure increases, baroreceptors are activated and send inhibitory impulses to vasomotor center in medulla oblongata. This causes decrease in blood pressure and inhibition of respiration. However, in physiological conditions, the role of baroreceptors in regulation of respiration is insignificant. Impulses from chemoreceptors. Chemoreceptors play an important role in the chemical regulation of respiration. Details of chemoreceptors and chemical regulation of respiration are explained later in this chapter. Impulses from proprioceptors. Proprioceptors are the receptors which give response to change in the position of body. These receptors are situated in joints, tendons, and muscles. Proprioceptors are stimulated during the muscular exercise and send impulses to brain, particularly cerebral cortex, through somatic afferent nerves. Cerebral cortex in turn causes hyperventilation by sending impulses to medullary respiratory centers. Impulses from thermoreceptors. Thermoreceptors are cutaneous receptors, which give response to change in the environmental temperature. Thermoreceptors are of two types, namely receptors for cold and receptors for warmth. When body is exposed to cold or when cold water is applied over the body, cold receptors are stimulated and send impulses to cerebral cortex via somatic afferent nerves. Cerebral cortex in turn, stimulates the respiratory centers and causes hyperventilation. Impulses from pain receptors. Pain receptors are those which give response to pain stimulus. Whenever pain receptors are stimulated, the impulses are sent to cerebral cortex via somatic afferent nerves. Cerebral cortex in turn, stimulates the respiratory centers and causes hyperventilation, Fig 126.3. Chemical mechanism. Chemical mechanism of regulation of respiration is operated through the chemoreceptors. Chemoreceptors are the sensory nerve endings, which give response to changes in chemical constituents of blood. Changes in chemical constituents of blood which stimulate chemoreceptors. Hypoxia, decreased PO2. Hypercapnia, increased PCO2. Increased hydrogen ion concentration. Types of chemoreceptors. Chemoreceptors are classified into two groups: central chemoreceptors, peripheral chemoreceptors. Central chemoreceptors. Central chemoreceptors are the chemoreceptors present in the brain. Situation. Central chemoreceptors are situated in deeper part of medulla oblongata, close to the dorsal respiratory group of neurons. This area is known as chemosensitive area and the neurons are called chemoreceptors. Chemoreceptors are in close contact with blood and cerebrospinal fluid. Mechanism of action. Central chemoreceptors are connected with respiratory centers, particularly the dorsal respiratory group of neurons through synapses. These chemoreceptors act slowly but effectively. Central chemoreceptors are responsible for 70% to 80% of increased ventilation through chemical regulatory mechanism. Main stimulant for central chemoreceptors is the increased hydrogen ion concentration. However, if hydrogen ion concentration increases in the blood, it cannot stimulate the central chemoreceptors because the hydrogen ions from blood cannot cross the blood-brain barrier and blood-cerebrospinal fluid barrier. On the other hand, if carbon dioxide increases in the blood, it can easily cross the blood-brain barrier and blood-cerebrospinal fluid barrier and enter the interstitial fluid of brain or the cerebrospinal fluid. There, the carbon dioxide combines with water to form carbonic acid. Since carbonic acid is unstable, it immediately dissociates into hydrogen ion and bicarbonate ion, Fig 126.4. Co and H2O rightward arrow H2 co rightward arrow H++ HCO3, hydrogen ions stimulate the central chemoreceptors. From chemoreceptors, the excitatory impulses are sent to dorsal respiratory group of neurons, resulting in increased ventilation, increased rate and force of breathing. Because of this, excess carbon dioxide is washed out and respiration is brought back to normal. Lack of oxygen does not have significant effect on the central chemoreceptors, except that it generally depresses the overall function of brain. 3. Peripheral chemoreceptors. Peripheral chemoreceptors are the chemoreceptors present in carotid and aortic region. Refer Chapter 101 for details. Mechanism of action. Hypoxia is the most potent stimulant for peripheral chemoreceptors. It is because of the presence of oxygen-sensitive potassium channels in the glomus cells of peripheral chemoreceptors. Hypoxia causes closure of oxygen-sensitive potassium channels and prevents potassium efflux. This leads to depolarization of glomus cells, receptor potential, and generation of action potentials in nerve ending. These impulses pass through aortic and herring nerves and excite the dorsal group of neurons. Dorsal group of neurons in turn, send excitatory impulses to respiratory muscles, resulting in increased ventilation. This provides enough oxygen and rectifies the lack of oxygen. In addition to hypoxia, peripheral chemoreceptors are also stimulated by hypercapnia and increased hydrogen ion concentration. However, the sensitivity of peripheral chemoreceptors to hypercapnia and increased hydrogen ion concentration is mild.